Much like the bone cells and other collections of similar cells that we've seen, neurons arise from a neural stem cell. However, this neural stem cell does not only give rise to neurons, it also gives rise to cells that are called glial cells, or more appropriately, they're now called neuroglial cells. These cells, they're like a support system for neurons. Neurons are so specialized in carrying action potentials and electricity that they pretty much don't like to do much of anything else. And it's these neuroglial cells that modify the interstitial environment in the brain. Some of these neuroglial cells, they also make a substance called myelin, which we'll discuss a little bit later on. Neuroglial cells make up about 50% of your nervous tissue. And there are different types of neuroglial cells that are found in the central nervous system compared with the peripheral nervous system. So for example, the cells that make myelin in your brain, they are not the cells that would make myelin in the nerves of your hand. One of the main neuroglial cells that are present in the brain, the most common one, are our astrocytes. They're so named because they are so frequent and because of their shape. Their orientation in space, it kind of creates this like structural framework for the brain, like the framing of a house. Your brain, or anybody's brain really, is actually about the consistency of tapioca, which I do not like. So it holds its shape, but it's definitely jiggly and squishy. Astrocytes, they are, their function is kind of like a server in a restaurant. An astrocyte will take nutrients like oxygen and glucose, they'll extract it from a blood vessel running in the brain, and it will feed that oxygen and glucose directly to the neuron. So it's not a buffet, right? In doing this, this prevents the neuron from being exposed to any chemicals or harmful substances in the blood vessels. Astrocytes can also help function in repairing damaged neurons. Current research on traumatic brain injuries is focused on astrocytes. There's a working assumption that encouraging their repair activities could be the key to recovering from traumatic brain injuries. Also, in your brain and spinal cord, there are these little tiny cells called microglia, and they're like feather dusters for your central nervous system. They basically roam around, kind of like those phagocytes in skin. They basically roam around and they clean things up. Technically, they do belong to a category of cells called phagocytes, which is that word that just means a cell that eats. So microglia, in roaming the central nerve system and cleaning up the debris, it's kind of, there's, again, a working assumption that microglia play an incredible important role in neural inflammation diseases. And it's believed that deadening their activity can actually deaden the damaging inflammation. But there's a trade-off there, because microglia are like the immune system of your brain. So deadening them can also bring about other autoimmune diseases that could have been lurking. Oligodendrocytes are cells that are involved in making a lipid covering called myelin. Kind of similar to the rubber covering on the outside of an electrical cord that prevents you from getting shocked if you were to touch that live wire underneath. Take a look at the oligodendrocyte here in this picture. It has these processes that are coming off of it and reaching out to two different neurons. And it's interacting with those neurons. And what you can see is at the end of its little arms of the oligodendrocyte are these pillow-type structures, which are the myelin covering that's wrapping around that part of the neuron. It's this structure, right, this little pillow foot, that gives them their name, oligodendrocyte, which literally means many branches. Many of us have heard about a procedure called a spinal tap. And there is spinal fluid that circulates between two layers of coverings around your brain and spinal cord. That fluid is very different than your blood. And sometimes that fluid needs to be sampled to rule out certain types of infections like meningitis. This fluid, it is called the cerebrospinal fluid, or we abbreviate it as CSF. This fluid fills all of the open spaces in your brain that are called ventricles, and those spaces are filled with cerebrospinal fluid. Those spaces are also lined with cells that are called ependymal cells, pictured down here at the very bottom. These ependymal cells, they have these little cilia on them, or these little hairs, that all wave in unison. And it's this waving action that circulates the cerebrospinal fluid through these open cavities in your brain and down your spine, and then back up to the area around your brain. These ependymal cells, they are critical in making the cerebrospinal fluid. However, they only do that at very few places in the brain. What they do, as you can see, because they're attached to a blood vessel, is they do something similar to astrocytes. They take what's in the blood vessel and make the CSF, whereas astrocytes take what's in the blood vessel and feed it directly to the neuron. There are only two types of neuroglia worth mentioning for us in this class that are in the peripheral nervous system. Everything that we have talked about before in this mini lecture belongs to the central nervous system. There are two types of neuroglia here in the peripheral system, and that means, right, that these two types of neuroglia are never found in the brain or spinal cord. Satellite cells are very similar to astrocytes in that they ingest and they regulate the interstitial fluid surrounding a neuron. They tend to wrap around the cell bodies of neurons in somatic tissues, and the satellite cells and the cell bodies, they form these clusters that are in your viscera, and we call those clusters ganglia. Similar to an oligodendrocyte, there are also cells that are called Schwann cells, and they make myelin sheaths for the same purpose that those oligodendrocytes made myelin in the central nervous system. However, Schwann cells make these myelin sheaths in a very, very different process than those oligodendrocytes make the myelin sheaths. The BBB, the blood-brain barrier, is a diffuse barrier. It's not like a membrane or covering or anything, right? It's not like tights. It's like fishnet stockings. What it is, it's really a collection of activity that's done by a lot of these neuroglia that are already present in your brain and spinal cord. So when we refer to the blood-brain barrier, it really is referring to a protective barrier for the central nervous system. By a bunch of neuroglia and other structures working together, they are able to tightly control the interstitial environment for neurons. Take a look at this picture, more toward the left of the slide, running down the middle of it, you have a blood vessel. So take a look at things more toward the left. Although we certainly want to get glucose and oxygen out of the blood vessel and to those neurons, there may be other things in the blood vessel that I don't want to expose my brain neurons to. So first, wrapping around a blood vessel are cells called pericytes. Not parasite as in blood-sucking parasite, but peri, P-E-R-I, word meaning around, and site, meaning cell. All right, so first layer of protection. Second, take a look at all of the green oligodendrocytes. which are pretty much like epithelial cells, just really big, simple squamous epithelial cells. They form a diffuse barrier all over the blood vessel, regulating what is allowed to leave. So third, take a look at all of the green astrocytes. They are touching the blood vessel and then reaching out to touch a neuron itself. They are regulating the interstitial environment. They are determining what gets to leave that blood vessel. And this picture here over on the right, it's kind of like a cross section or an inset of a blood vessel. I mean, look, there are layers that molecules would have to go through in order to enter into the interstitial environment. There's the pericyte, the endothelial cells of the blood vessel, and then there's also these astrocytes. And then once you get through all three of those layers, you also you have to kind of be lucky and not get cleaned up by a feather duster, right? You'd also have to get lucky and escape detection by a microglia. So all of these cells, they all work together to form what we call the blood-brain barrier, right? So all of these cells basically have the ability to prevent the intrusion of things from the blood into the very closely regulated interstitial fluids of the brain. And one example that some of you may know about is ketoacidosis, which is a state that a diabetic person might get into with poorly regulated insulin and blood glucose levels. Basically, why is ketoacidosis so bad? Because it creates ketone bodies from fat deposits, and ketone bodies are able to escape the detection by the blood-brain barrier. Sometimes this barrier gets it wrong. And what happens is those ketone bodies, they're able to actually get into the interstitial environment, and those ketone bodies eat away at your neurons, kind of like an acid eats away at a protein. So the blood-brain barrier, it's not like 100% guarantee that something won't get in there, but it certainly is a pretty substantial barrier that can prevent intrusion.